So I'd like to welcome you all to the C4C Center for Commercialization Entrepreneurial Faculty Fellow Lecture Series. My name is Jennifer McCullough. I'm a Senior Technology Manager at the University of Washington C4C. And while still new to this particular portfolio, I'm honored to have the pleasure of introducing our Entrepreneurial Faculty Fellow, Dr. David Baker. Dr. David Baker is Professor of Biochemistry and Adjunct Professor in Genome Sciences, Bioengineering, Computer Science, Chemical Engineering, and Physics. His research group develops methods to predict three-dimensional structures of proteins, as well as their interactions with other proteins, and also works on designs and validations of novel macromolecules with new and targeted functions. David has the distinction of being an investigator of the Howard Hughes Medical Institute and a member of the U.S. National Academy of Sciences. He started at University of Washington in 1993, and it was shortly thereafter that he sent his first disclosure to our office in 1997. Since then, he has submitted over 45 invention disclosures, one of which is the Rosetta software suite. Rosetta currently has active licenses with over 23 companies, including many world-class pharmaceutical and biotechnology companies. David was selected as an entrepreneurial faculty fellow not just because of his successes with commercial licensing of the Rosetta software, but also this software serves as the foundation for two startup companies, Arzeta and Bioarchitecture Lab. It's also the foundation for three other startup companies that are up and coming through the Center for Commercialization. So with that, I'd like to turn the podium over to Dr. David Baker to tell you about his commercialization experience, translational protein design. Please help me welcome Dr. Baker.
Well, thank you for that very nice introduction. Can you hear me? So I'm going to give you first a few minutes of introduction on protein design so you know what it is, and then talk about translation and commercialization. So the idea of protein design really comes from looking out in nature and into ourselves and seeing all the marvelous proteins that basically are responsible for all the critical processes in life. And it's really kind of remarkable how well living things sort of solve the challenges of capturing energy, moving around, even thinking, and so that shows the power of selection operating on proteins to shape them to carry out functions that are very sophisticated, and sort of the idea for protein design is that we now face challenges that weren't faced during evolution. So while we have all these naturally occurring proteins that solve all the problems that arose during evolution really, really well, we face new problems today, ranging from health problems that come about because we live longer, new types of infectious diseases, like the one I might have now, so stay away. We're heating up the planet, we need new sources of energy. All these sorts of things are problems where, if we look out in nature, we think that it appears to be a good chance that proteins could be part of the solution. And so really the protein design problem is to develop methods for designing a whole new world of synthetic proteins that address modern day challenges. And you can sort of see the link immediately to translation, commercialization, because we're trying to develop methods for designing proteins that do things that are useful. And if you make something that's useful and you want it to actually be used, you have to get it out in the real world, which means you either have to develop it and then license it to a company, or you have to start a company yourself. Okay, so the way, again, I'll spend a few minutes talking about the way this work actually is carried out. So we've developed a computer program called Rosetta over the years, which Jennifer alluded to, for calculating amino acid sequences which are optimal for the desired function of the protein. Since we designed these proteins, we know they're amino acid sequences, and what really enables all of this is the fact that DNA has become essentially a commodity item. There are many different companies that manufacture DNA. Any amino acid sequence you come up with, you have a choice of 5 different companies who will get you that piece of DNA in anywhere from a month to a day after you order it. Once we get the DNA back, we go from the computer to the bench, we make the proteins and assay them and see if they do what we designed them to do. And just a couple animations to illustrate how this works. Say this is a protein backbone. If we want to, a design calculation looks something like this, where different amino acids have, each of the 20 amino acids is the same backbone, but a different side chain coming off of it. And this just shows a Rosetta calculation. We're trying to find the amino acid sequence, that combination of side chains, which has lowest energy in this backbone. Then, once we have identified a sequence which is very low in energy in a backbone like that one, we then have to see whether that sequence actually is predicted to fold up to that structure, not fold up to some other structure that might be still lower in energy. So over the years, we've also developed in Rosetta methods for predicting how proteins fold. And so here you can see a protein folding calculation where we've taken, we take design sequences, predict what their structure is, and then if they're predicted to fold up to exactly that structure we designed, then we go ahead and order genes. So if we have a sequence that we've designed a sequence using the calculation in the movie I just showed, we check that sequence to see whether it's actually predicted to fold up to the structure we want in the calculation I just shown here. If a sequence passes those two checks, then we order a gene that encodes that amino acid sequence. And this is one such example where we are simply trying to design a sequence that would fold up to a particular structure. So the structure is shown here. In these calculations, which are very compute intensive, we make use of volunteers all around the world. So this is, each red dot is the contribution of a different volunteer somewhere in the world. We have about 350,000 volunteers now. This is a project called Rosetta Home. And so we send out the sequence for this protein, and each red dot is the lowest energy structure that that volunteer found. And this is the energy, and this is how far that structure is, the one they found, from the one that we are trying to design. So what you can see here is that people are finding solutions all over the map in terms of energy and distance from the target structure, but there are the lowest energy solutions that anyone finds are very close to the structure we were trying to design. So in that case, we go ahead and make the gene, and what we find when the design works is that when we experimentally test it, that the protein folds up to essentially the identical structure. And in this case, this is just a protein that is designed to have a particular structure, but the same thing goes if it has a particular function. So one of the, so just having proteins that fold up to structures is interesting, but one of the obvious applications is binding. So we want to design proteins that will bind to targets. And one of the ones that we've been looking at for a while is the influenza virus hemagglutinin. This is the protein on the surface of the virus. And there are two regions which are really critical to the function of the virus. This region here, where it binds to receptors on the surfaces of our cells. And this is another site which is very conserved between different viral strains. So we've used the techniques I described to design proteins, small proteins which bind very tightly to either this site or this site. This is sort of a blow-up of what this site looks like in the virus. This is the surface of the viral protein in yellow, and these are two small proteins that we've designed which fit together in a very shape complementary way onto the surface of the virus. And when we make these proteins, we find that some of them, when we test them experimentally, most of them don't actually bind the virus, but a small number do. And when it's experimentally determined how they bind the virus by X-ray crystallography, we find that they bind the virus in essentially exactly the way that they were designed. So this is the viral protein here. This is the design. One of these is the crystal structure, experimentally determined structure, and the other one is the design. And they really come out exactly the way they were supposed to. And this is a design that Eva Strauch made to the head of the virus. So we can design binding proteins to different places on a pathogen protein. Now there are a couple different applications of binding proteins. One of them is to diagnostics, and we have an exciting collaboration with Carly Holstein and Paul Yeager, who are developing paper-based diagnostics. And basically what Carly's doing is putting one of these proteins down on a piece of paper, on a strip, like on a pregnancy test, and then the virus gets captured, or the viral protein gets captured, and then she can read out with another one of these proteins, like this one. And that's looking very promising. And so you get a nice sharp band when there's virus present on the piece of paper. Another application, which is a great collaboration with America Trins and Deb Fuller at the UW, is therapeutics. And America and Deb have some very exciting data now showing that if mice who are infected with the flu, if they're given one of these design binding proteins, it actually completely protects them from losing weight and from dying. So the mouse gets the flu, they start to lose weight, and then they eventually die around here. If they get the binder protein at the same time, they're totally fine. And America and Deb have found that you can get treated with this protein 24 hours before or afterwards, and it doesn't really matter. So that's one of the things that we're excited about, that starting to think about or actually more than thinking about starting a company based on. And this is like a lot of things in the lab where we didn't really think about it being a company to begin with, but then you get results like this and you think, well, maybe this is ready to go. So there's still a number of steps before that can happen, but it's pretty exciting. So another example of translating protein design into the real world, this is more work by Eva Strauch. There's a lot of interest in the biotech and pharma areas in purifying antibodies, because a lot of drugs are antibodies. And there's a protein called protein A, which binds antibodies, and it binds. This is a binding curve. So this is showing how much antibody is bound as a function of its concentration. And so this standard protein called protein A binds antibodies very tightly, both at high pH and low pH. The protein that Eva's designed binds tightly at high pH, that's shown here, but at low pH it completely comes off. And this isn't very low. This is pH 5.5. So that's interesting, because it means if you want to purify antibodies, you can purify. It binds you can purify them at the higher pH and then elute them under very gentle conditions, which is important because a lot of proteins denature at the very low pH, which you have to use with protein A. So this isn't. So I've sort of talked about a diagnostics application for design proteins and a therapeutic application. Now this is sort of an application as a molecular reagent. And in this case, we're kind of going a different route. We're sending this protein out to pharma companies who are trying it out in-house with an agreement in place where they sort of give us feedback on what they're finding. So along with Rosetta Home, which involves the general public, we've also developed with colleagues in the computer science department a game called FoldIt, which lets the general public get involved. And the FoldIt players work on whatever we're working on. And so they've designed flu binders, for example. One of the things they're working on is a protein that binds the A-beta amyloid protein that's implicated in Alzheimer's disease. So this shows a really neat FoldIt design. They start with a puzzle. Looks like this. They build something that completely wraps around the beta peptide. So we're having a lot of fun, and it's a good way of getting the general public sort of knowledgeable about some of the molecular mechanisms. And who knows, maybe something useful will come out of this. All right, so now I'm moving on to the more commercialization part of the talk. There's a big development community. One of the things that we did early on that I think was really a good idea was we decided that no one was going to get any money in their pocket from developing Rosetta, because it was really a team effort from the beginning. So all of the proceeds we get from these companies who license Rosetta go back into a common pool that then pays for an annual meeting where all the developers get together. It supports research. We have a little granting agency now that people can apply for developing new aspects to the code. And then this also supports a core team of a few people who manage the software, new releases, dealing with integration of different things. It's a bit of a challenge because we have people who are now developing Rosetta really all over the world, not just in my group, but in all the groups that have left my group. I mentioned already crowdsourcing of computing and brain power. We're pretty excited about this because it ties together a lot of strengths of Seattle. Biomedical science, obviously Seattle's very strong in this. Software, gaming, again, Seattle's great for the software industry, and obviously health is a big issue in Seattle too with the Gates Foundation too. So we're, as Jennifer mentioned, there are a number of new companies in the works, and Ingrid's here. You've probably, I hope most of you had the good fortune of hearing her talk. So Ingrid, I should make you come up here. Where is she? She's hiding. Okay, so that's Ingrid. Stand up so they know who you are. I told her I was going to embarrass her if she actually came. Anyway, so she has a really exciting enzyme that you should talk to her about after I'm done. That breaks down gluten, and it's really a promising potential therapeutic for celiac disease. And so Ingrid's started a company, and she's working now. I'm sort of doing things in reverse, but we were very fortunate recently to get a grant from the Life Sciences Discovery Fund to allow people like Ingrid to stay in the lab a little bit longer, to do what's needed to be done to develop protein designs to the point they can be useful in the world. And so Ingrid's making her protein so it'll really degrade gluten fast, and she's hoping to start a company, spin out of the U within the next year. Another area that's being pioneered by Neil King, who's here, is designing self-assembling particles that you can put things in for targeted delivery. So you could put RNA inside, you could put small molecules inside, and or proteins inside. And so Neil's trying to develop this as a general platform for targeted delivery. Another area that we're very interested in is you can put things on the outside to make very custom designed vaccines. So this is sort of a schematic of how it works, which I won't go through. But when a lot of times you do this stuff first, and then you sort of start thinking about all the different applications afterwards. So for example, the vaccine application didn't really come about till we actually had these particles and realized they looked something like viral particles. So another area in the lab is to design proteins to bind small molecules. So there's a number of different applications to proteins that bind small molecules. For example, diagnostics. There are a lot of these molecules you'd want to detect, like the state of Washington is very interested in detecting THC. Oh, apixaban is an interesting one because an overdose of apixaban, which is a drug that a very potent drug for a blood thinner. If you get too much, it can be very dangerous. So if you have a protein that soaks it up, it could be useful. So we've got binders to a number of small molecules. We're making more, and each of these potentially could be the basis for a spin-out company or or simply a license something. So it sort of shows kind of how open-ended protein design is. You can, when you make new things, if they're useful, then there's it's kind of neat. There's kind of more people in the lab are now excited about sort of actually taking things out into the real world. So there are a lot of different projects in the lab that would have direct translation. These are ongoing projects which are interesting science, but once they work, then there's sort of an interesting opportunity for translation. And so I mentioned this already. The idea of taking things that are basically straight out of a scientific paper and then trying to nurture them in this institute we started, the Institute for Protein Design, with support from LSDF and philanthropists. We can now mature these further on to the point where they can be spun out. We're very excited about a program that is just starting up. I think the announcement just went out today. The Washington Research Foundation has funded an Innovation Fellows Program. And the basic rationale for this is, in a lot of these, there are a lot of different applications for protein design. And in all of them, you sort of have to be expert enough to identify what an important challenge is, what would be something very useful that you could make, a reagent or diagnostic or enzyme. And then downstream, you have to have the expertise to test it say, if it's an anti-cancer agent, you have to be able to test it in an animal model. And while we're good at designing proteins, we aren't very good at the upstream part and the downstream part. And so this program is now, we're trying to leverage all of the great expertise in the Seattle area. So the basic idea is that PIs throughout, basically anywhere in Seattle, can propose projects. We're nationally advertising. People can go into any of these labs and apply protein design to the area of interest, and then hopefully that'll lead to a lot more company formation activity down the road. It looks like I've run out of slides. So the acknowledgement slide, I'm sorry, I seem to have left out, but I obviously didn't really do any of this myself. I've been very lucky to have fantastic collaborators, and now let's get to the more important part. Are there any questions?
How do you see big data with genomic information being transferred to a system like yours with protein? Do you ever see a combination of the two meeting, or do you always see them kind of being separate?
No, I mean, there's a lot of big data in biology, like genome sequence information is a big data problem, right? And so we leverage all the amino acid sequence and all the DNA sequence information that's out there, both in terms of getting ideas for starting points for designs, and then often for, we can learn from them to try and improve the design process overall. So there's quite a match there, in fact.
Yeah. How do you get the word out to the general population about Rosetta? How do you get them interested in it and involved?
Yeah, well, so we were very lucky. Before we started Rosetta at Home, SETI at Home had been in existence for a number of years, and SETI at Home is the search for extraterrestrial life. And the people who developed SETI at Home, primarily a guy named David Anderson at Berkeley, realized they had developed this really powerful resource for infrastructure for getting the community involved in computing, and they were interested in applying it to biomedical problems. So we contacted them. They really helped us get Rosetta at Home up and running, and then we had immediately people who were familiar with distributed computing, like SETI at Home participants who were some of the early Rosetta at Home participants. And then Rosetta at Home, and similarly, was the sort of a lot of the source for a lot of the people who played FoldIt initially.
Do you use any social media to keep in contact with each other?
Yeah, so there's a lot of social media connected with both Rosetta at Home and FoldIt. Some of which we do, like for example, I have essentially a blog on the Rosetta at Home site, and we give feedback on the FoldIt, but it's just we don't really have the bandwidth. So what's really been effective is getting players and participants to talk to each other. So like FoldIt has a very active chat. There's always stuff going on. Some new person will start. They'll say, well, what do I do? And then 5 people will answer them and try and recruit them to their team if they look good, so.
At the very beginning, when you get the gain out of the protein by back translating it, so how do you take care of the degeneracy?
Yeah, well so it depends on where you're going to make the protein, and different organisms have different preferences for which codons. So you may have 4 different codons for a particular amino acid. So you try and pick the one that's most common for that organism where you'll make it.
Can you share any information about the companies that are using the Rosetta software, successes they've had in using it for development?
Well, I can't because they don't tell us. I mean, I think the reason why they license the software and what they do with it is state secrets or company secrets, really. Yeah, Deb.
So I'm just curious, let's say you get a gamer that discovers some protein that could cure some disease.
Like
what's
flu,
the
for
next,
example.
Yeah. What's the next step then?
Well, I think it basically, like everything we do, it's all owned by the UW. So the gamer would, let's say they make, they make an amazing new flu binder. So we would file a disclosure with C4C, with the UW. The gamer would be listed as an inventor. They would have all the rights that inventors, like you or I would if we invent something at the UW. And, so I think.
100,000 hit points right away.
Yes, exactly, that's right. Yes, that's right. They get more than we do because we'd probably make a big, hoopla about it, right. Yeah.
So many different ideas, different companies, products. How do you
Yeah, that's a really good question. A lot of it depends on whether there's someone like Ingrid who's interested in starting a company. So I think it's fair to say, Ingrid, if you hadn't wanted to start a company, it would have just been licensed. So if there's someone who's involved in the project who wants to stay on and start the company, then it goes the company route. Whereas if there isn't, then it would go the license route. That's sort of how it's breaking down now. Yeah.
So you guys, your department's always done design. It may not be the right place to ask. There's other methods using evolution where you try different.
Yeah.
You try to evolve cycles of design and evolution. This might not be the right place to ask, but do you got, I mean, do you do that too?
Yeah, we do that too. Right, right. I brush that under the rug, right? Because so for essentially everything that we've made that's been successful, I mean, with design, we get in the ballpark. So I showed you that those little flu-binding proteins that were binding exactly where they were supposed to. But the things that the actual amino acid side chains that we had in the design weren't all optimal. So it turned out we were able to make small changes, just change 4 or 5 amino acids, make slight changes, and we got a much, much more effective binder. So what we always do after we get the initial, so we order a bunch of genes, they come in, we test them, we take the best one, and we make small numbers of, basically we make all single amino acid changes and select out the ones that work the best. So we're always getting, that's good for a couple reasons. First of all, it makes the function, whatever we try to design makes it better. And second of all, we get a lot of feedback for improving the computational model, which is pretty far from perfect. That's why it's almost always possible to find substitutions which make it better.
And thank you all for coming. Please help me thank David again.